Okay, this is part two of DNA, genes, and chromosomes. Welcome to Let's Get to the Marks. Let's get into the vid. So, question four. Complete table one to show three differences between DNA in the nucleus of a plant cell and DNA in a prokaryotic cell. So we've got table one here. Pause the video, grab a pen, write it down on a scrap bit of paper, then unpause and check your answer. Okay, let's hit it. DNA in the nucleus of a plant cell. What can we say? Well, plant cells are eukaryotic, so it has histones. Really, it's attached to histones. And of course, this has no histones. So DNA in prokaryotes have no histones. DNA in a plant cell is longer. Um, it's going to be shorter in a prokaryotic cell. They don't require as many genes. The DNA in a plant cell is also linear. What do we mean by that? Yes, our DNA is in a double helix, but it's not attached at the ends. It's in one piece. The end's not attached. However, the DNA in a prokaryotic cell, it's in a loop, or it is called circular DNA. There's a couple of other things that we can mention. These are all alternative answers. Plant cell has introns. Remember the junk DNA, the non-coding DNA. A prokaryotic cell only has exons. It only has coding DNA. So there's a couple of other little things there. We could also say as well, eukaryotic cells have no plasmids. Now, plasmids are tiny, don't get them mixed up with circular DNA in prokaryotes. Plasmids have only got a few, four, five, six, whatever genes on them. They're tiny, tiny circles of DNA. So you could have said that prokaryotic cell has plasmids. So I've given five answers there for each. And of course, you only need three for each, and there's 3 marks. Beautiful. Now, scientists investigated the genetic diversity between several species of sweet potato. Why? Well, they've got nothing better to do. They studied non-coding multiple repeats of base sequences, defined non-coding base sequences, and described whether non-coding multiple repeats are positioned in the genome. So these non-coding repeats are known as introns. And basically, you're looking at bases or DNA that does not code for amino acids. You could say polypeptides. So DNA that ultimately does not code for a polypeptide, or DNA does not code for amino acid, same kind of thing. Now, the second mark is, and where do you find these repeats? So um, they are found in introns. So you only really get these in eukaryotic cells like sweet potatoes, which is a plant. So we find them in introns. We can say between the exons or coding um, DNA. So you're going to find them in between the genes that code specifically for amino acid sequences and polypeptides. You're going to find the introns in between those. Okay, so coolio. So you get 1 mark for saying define non-coding DNA, so it doesn't code for amino acids. You could have said all polypeptides there. And then where do you find them? You find them in junk DNA called introns. You find them in between the exons, between the coding DNA. Awesome.
Keep moving on, keep moving on, keep moving on, keep moving on.
All right, we're on question five. So, question 5A, the nucleus and chloroplast of a plant cell both contain DNA. Give 3 ways in which the DNA in a chloroplast is different from the DNA in a nucleus. This is quite interesting because this ties into a theory called endosymbiosis. You may be interested to look up on that theory about the origin of chloroplasts and mitochondria within eukaryotic cells. Anywho, skipping on to that, let's get down to it. So think of this question when they start talking about DNA in chloroplasts. It's basically the same as the DNA you find in prokaryotes, which gives us a link to what a chloroplast was in a past evolutionary life many, many years ago. So how is it different from the DNA found in a nucleus? So what do we want to say? We've already said them. It has no histones. Of course, the DNA in a nucleus has histones. So has no histones. We're going to write the same 5 things down, right? Um, or 4 things. Has no histones, has no introns. What else can we? So it's got no junk DNA. It has circular. DNA. So the chloroplast has its own circular DNA, even though the rest of the plant cell has linear DNA. The nucleus of the plant cell contains linear DNA. The chloroplast has circular DNA. That gives us a hint to its past, evolutionary past. So there we go. I've mentioned three things. Ah, lovely jubbly. And then we could probably say the DNA inside the chloroplast is going to be shorter because it contains fewer genes than the DNA in the nucleus, which contains the genes for the whole of the plant, whereas the DNA in the chloroplast is just the genes for the chloroplast. But there you go. Any two of those three will do. Oh, you had to say three differences. Well, it's lucky I gave three differences, right? So there we go. I gave three differences anyway. That's me thinking it was a two marker. B, some DNA nucleotides have the organic base thymine, but RNA nucleotides do not have thymine. RNA nucleotides have uracil instead of thymine. You know, give one other difference between the structure of a DNA nucleotide and the structure of an RNA nucleotide. So what's the other difference? Think about your nucleotide. Both contain a phosphate, both have a pentose sugar, both contain bases. However, we've just been informed the RNA base has U instead of T. What's another difference? Well, it's right here. It's not in the phosphate, it's with this pentose sugar. What's the name of the pentose sugar in RNA? So we would be like RNA has ribose sugar, whereas DNA nucleotide has deoxyribose sugar. And that's really the only other difference with the nucleotides. There's only two differences. One is the uracil base, and the other is on the sugar, there's a difference. However, there is difference between RNA and DNA, as in DNA is double-stranded, RNA single-stranded, etc. But this was specifically about the nucleotides, not about the strands of RNA and DNA. Hokey cokey, let's move on. The diagram shows the tRNA molecule. So this is a transfer RNA molecule. And we're going to be asked name structures W and X in the diagram. Well, I'm pretty sure you'll probably remember the structure X. This is what binds by complementary base pairing to the codon on mRNA at the ribosome. CUC actually codes for leucine, the amino acid, and it is known as the anticodon. So this is the anticodon. It's where the tRNA molecule temporarily hydrogen bonds or binds by complementary base pairing to the ribosome. Now, name the structures labeled W and X in the diagram. So W, what is W? What attaches to W? Well, W is where it's going to carry the amino acid. Obviously, this will, the tRNA will bugger off and leave leucine behind, and leucine will be bonded by a polypeptide, by a peptide bond to another amino acid. So this is being brought down. What we call W is the attachment site or the binding site. So it's the amino acid binding site. It's where the amino acid attaches or binds or bonds to the tRNA. Of course, the amino acid is left behind when tRNA leaves, as the next tRNA molecule comes. Okay, and that is going to get you a mark. You needed both to get a mark. That's quite difficult, actually. I guess anticodon everyone would have got, but the amino acid binding site may have slipped a few people up. Right, let's go down. Let's finish this off. So part D, not all mutations in the nucleotide sequence of a gene cause a change in the structure of a polypeptide. Give two reasons why. So why a mutation is a random change in one of the bases. So G could become C, C could become T, but then you get deletions where, say, T will disappear or G will disappear, and then you get insertions where extra bases are put in. But what kind of mutation will mean that nothing changes? The polypeptide retains its 3D shape and is still functional. So what would that be? And of course, we could get a silent mutation. What is a silent mutation? This is, remember that, I'm just going to put the explanation. DNA is degenerate. DNA is degenerate. So each amino acid has more than one code, right? So each amino acid has more than one codon. So changing the triplet code may not change the amino acid. Let me give you an example. So for example, just so you can understand it, CCC codes for glycine, right? Imagine there's a mutation. One of the C's is turned to a G. CCG also codes for glycine. So essentially, we're going to get a mark for saying, well, just because there's a mutation, it might be a silent one. There's more than one triplet for each amino acid, and that will essentially get you those 2 marks. Lovely. Now we need another reason. Where could the mutation happen where it wouldn't affect the amino acids? Of course, the mutation could occur in non-coding DNA in introns. So if it occurs in the non-coding DNA, that's not going to do anything. So if the mutation occurs in non-coding DNA, they do not code for amino acids. So it's not going to affect it. So we have to say it could occur in introns, and these don't code for amino acids. Boom, we get all the marks. All right, we're nearly there, folks, nearly at the end of this pack. Question 6, compare and contrast the DNA in eukaryotic cells with the DNA in prokaryotic cells. Oh my gosh, how many times are they going to ask this in this pack? It's kind of good revision though, right? So we want to compare and contrast. We've got to talk about both. We can't just talk about one. We'll only get half the marks. So compare and contrast the DNA in eukaryotic cells and the DNA. Now, the fact that they say compare, that means what's the same, contrast, what's different. So we want to say, DNA in eukaryotes, oh my gosh, eukaryotes is linear and longer than the shorter and circular DNA in prokaryotes. There we go. I mentioned 2 lovely things there to get me some marks. What else can we say? Also, let's talk about some other differences. DNA in eukaryotes is attached or wound around histones. So it's if you can't remember histones, it's attached to a protein. DNA in prokaryotes is not. So I think I've done enough differences there to, you're going to need like 3 differences and 2 similarities here, because it's basically saying what's the same, what's different. That's enough differences. I could also say, we could also just, let's do the rest of them. DNA in eukaryotes found in plasmids. Sorry, not found in plasmids. Anyway, I'm not sure this one's a very good point, but there we go. And then we've got the no introns. So introns in eukaryotes, no introns in prokaryotes. Now, did I need to say all of these? No, this would probably, even though it looks like 5 marks, would probably only get me 3 marks, because they've actually said the word compare. What's the same? Contrast. What's different? So the same and different. So what is the same? What do they have that's the same? So both contain pentose sugars. Let's just list off some things. Phosphodiester bonds. I could explain the location of those, that the phosphodiester bonds are between the sugar and the phosphate. Phosphate groups. What else can we say? Both contain the bases GC and A. GCAT, sorry. So both contain the bases GCA and T. So here's a lot of similarities. They both got pentose sugars. They both have deoxyribose. I could say deoxyribose. They both have phosphodiester bonds. They both contain phosphate groups. They both have GCAT, and they're both held together by hydrogen bonds. So held together by hydrogen bonding. So there's loads of similarities as well. You could just pick a few of those. And they'll smash you out the rest of the marks. All right, let's move on. We're nearly there. This is the last question. So question 7, let's fit it on the page. Figure 1 shows chromosomes present in one human cell during mitosis. So we've got figure 1 and then figure 2. In figure 2, the scientists arranged the images of these chromosomes into homologous pairs. So, okay, so they've arranged them into homologous pairs. The dark stain used on the chromosomes binds to some areas more than others. That's why we get this striped appearance with like dark black light, dark black light. Okay, suggest one way the structure of the chromosome could differ along its length to result in the stain binding more in some areas. So why is some of the parts more heavily stained? We can just say you're going to get more bases there. Okay, it depends on the density of the bases in that region. So we could say more super coiling in some regions. So there's more super coiling. Depends on how tightly wound around the histone in each part. So some parts of the chromosome may be more like wound more tightly around the histone. So you're getting more bases and it's more base dense in that region. You get the idea. Anything that's going to explain that. So as to that, there's more bases in certain parts of the chromosome because it's more tightly wound around the histone, or there's more coils, more supercoiling. Okay, let's zing on. Okay, in figure 2, the chromosomes are arranged in homologous pairs. What is a homologous pair? So a homologous pair is like when you have in your cells, in your nucleus, you have a chromosome from mum and a chromosome from dad. Remember, I talked earlier about in part 1, I talked about chromosome 15. So, for instance, we look at chromosome 15. Every human being has a chromosome 15 from their dad, which is the paternal, and one from their mum, which is the maternal. And on the exact same loci, which is the position on the chromosome, we're going to find the exact same gene at the same position on the chromosome. So, for instance, for eye color, you could get big B, or big B, little b. You get different versions of the gene, but they're the same gene with slightly different versions called alleles. So you don't get the same allele, so don't make that mistake. So, anyhow, let's explain this. What is a homologous pair of chromosomes? Two of the same chromosomes, maternal and paternal, um, each with same genes in the same locations, same loci, um, may have different alleles in the loci. Didn't need to say that actually. All we really have to say is same gene. It's a chromosome with the same genes in the same places, same genes in the same locations, same locus, or two chromosomes with the same loci. There we go. But don't say that they have the same alleles. That could be a big misconception. They don't have the same alleles. They've got identical genes and the genes are in the same locations, but they could be slightly different versions of the genes, which are known as alleles. Okay. Sing on, sing on. That's the end. Don't forget, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.